guys, welcome to Learn Extra Live in 2013. Today we are doing life science for grade 12 with Aslam. How are you, Aslam?
Hi, Katlego. Nice to see you again.
Nice to see you too. Happy New Year.
Same to you, darling.
Thank you. So guys, what are we doing today?
Today?
Well, Aslam, what are we doing today? They don't know what we're doing
They
today.
will not know. Today it's all in the genes.
Oh, it's all in the genes.
That's it.
What kind of genes are we talking
we
about?
talk
here?
about that just now.
Okay, guys, so as you heard, we are doing genes today. It is awesome, guys. It's awesome to have you here today. So make sure that you're fully tuned in. For you grade 12s, you guys are now grade 12s. I know I've been saying this every show, but time has literally flown by. It's now grade 12, guys. So much work goes into this year. Therefore, so much more to learn extra with you guys this year. Our Facebook page, facebook.com/learn extra. And our Twitter handle is @learnextra. Guys, make sure you join us. We need your everything that you have. Just place it on the page, guys. And of course, our show is sponsored by Macmillan. Thank you, Macmillan, for sponsoring this grade 12 show. You guys are awesome. Where would we be without you? You grade 12s, obviously by now you know we have a website, right? But this time it's so much more awesome because what it has your notes, guys. It has life science notes right now. So go check it out. I will post it on the page. So you guys don't need to worry, but go check out the notes, videos, questions, everything that you need to know is on the page, guys. Please, please, please call all your friends. They need to join us right now. It is so exciting, guys. I'm excited. I don't know about you guys. Aslam, are you excited?
Very excited.
Over to you.
Thank you, Katlego. Guys, if you didn't hear the excitement in her voice by now, then there's something wrong with you. Welcome to 2013, guys. I can't believe we've already begun. Today we're talking about genes, guys. No. Not this one here that's on my neck. This is the one you wear. We're talking about the genes that make you, not the ones that you wear. Katlego.
Okay.
All right? So what is this all about, this genes? And the genes that we are talking about is obviously spelt G-E-N-E-S, not those ones. These are the ones that you wear. These are the ones that make you. And where are these genes situated? They are situated in the DNA. Let's look at that in a little bit more detail. Now, what are we going to look at today? Firstly, we're going to look at the location of DNA. And location in this sense does not mean a township, Katlego.
Okay.
It means where would we find the DNA? Where is the location? Where is the township of the DNA in the cell? That's what we're talking about. Okay? And we talk about chromosomal DNA and mitochondrial DNA. We do mention DNA that's found in the chloroplast as well, but we look at these two in more detail. We also want to know, how did we come to know about this DNA? In other words, we want to know the history of the discovery of the DNA molecule. Further, we want to know about this DNA and the RNA and what they're made up of. They are made up of building blocks called nucleotides. And we go further, the three components of a nucleotide, and they are the nitrogenous bases. And there's the names given to you. We're going to look at the names in more detail later. We talk of the sugar portion and the phosphate portion. So basically, guys, there are three main components of the nucleotide, and they are the phosphate, the sugar, and the nitrogenous base. And incidentally, notice I went the other way now when I was talking about it. That's the order in which they occur from outside in. Phosphate on the outside, followed by sugar, followed by the nitrogenous base. We then move on to the functions of DNA. Why do we have DNA in our body? And lastly, we look at DNA replication. This is for today's lesson. This is what we are looking at. Let's go into the actual work then. So the first question we asked ourselves is, where do we find this DNA? In other words, what's the location of DNA? Let's keep with the vocabulary, guys, and as we're going along with the lessons, every new term you get or you come across that I'm mentioning or it's on the screen, take the word down at the back of your notebook somewhere and build up this vocabulary, because terminology in life science is key to whatever we're going to do. So we're saying, where do we find the DNA? And we say that we said earlier, there are two kinds of nucleic acids. We get deoxyribonucleic acid, DNA, and we get a ribonucleic acid, that is RNA. These are the two nucleic acids that we have. DNA is the one we're looking at the moment, is found in the nucleus of eukaryotic cells, where it forms part of the chromatin. Some also found inside the mitochondria and the chloroplast. Now, notice that we're talking about, the other words are more or less simple, but we're talking about this guy here, eukaryotic cells. What is a eukaryotic cell? A eukaryotic cell is a cell that has what we call a true nucleus. In other words, a nucleus that is surrounded by a nuclear membrane, and inside we find the nuclear material. That means chromatin network, the nucleolus, etc. Whereas a prokaryotic cell is a cell, what we would call before, first. And these are what evolutionists and scientists would call the primitive cells. These so-called primitive cells lack a nuclear membrane. They lack a true nucleus. They have nuclear material which is not within an organelle. They are found in a cell. In other words, there is DNA or RNA or both, but they are not bound by a membrane, and they're not in an organelle. So the eukaryotic cells are the cells that after, not okay? Where it forms part of the chromatin, right, the chromosomes, and some is also found in the mitochondria and some in the chloroplast. Mitochondrial DNA, chloroplast DNA. We are very interested in this DNA that's found in the nucleus, and later in evolution, we are also interested in the mitochondrial DNA. But as we progress with the work on DNA, as you're going to go along, you'll notice we'll talk more about mitochondrial DNA. So we know now where it's found. Now we want to know how is this thing structured. In other words, what is the structure of this organelle? The first statement there, again a big word, guys. Remember, write the big words down. Polynucleotide. Nucleotide we've already discussed. Nucleotide is a building block of the nucleic acid, DNA and RNA. Both are made up of nucleotides, many nucleotides. If we look at this nucleotide, we can consider the nucleotide to be like a brick on the wall. The wall is made up of several bricks. The same way, DNA is made up of several repeating nucleotides, maybe with similar nitrogenous base or with different nucleotides. Each one has its own sequence. So polynucleotide, many. Poly means many. So nucleotide, poly, many nucleotides. So it means it's a large molecule. A polynucleotide is a very large molecule made up of a string of repeating units called nucleotides. We've discussed that already. Each DNA nucleotide consists of 3 parts. And I told you we're coming back to this now. 1, deoxyribose sugar. Remember, in DNA, we are talking about deoxyribose sugar. We must understand this because when we talk of RNA, we're going to talk about a ribose sugar. So keep that in mind. 1 phosphate group, and it's not phosphorus, but it's a phosphate group. Keep that also in mind. And 1 nitrogen-containing base. There are 4 possible bases. What are they? Adenine, A, thymine, T, guanine, G, and cytosine, C. Those are the 4 types of nitrogenous bases one would expect to find on a DNA molecule. Okay? Now let's look at the structure. There we have it. phosphate there, attached to a deoxyribose sugar there, and that is attached to a nitrogenous base. This is one nucleotide. Represents one nucleotide. Now let's look at that in a molecule. There we go. In other words, we're looking at something like this. There, in color, there's it. We're taking this guy out, and we are putting it there. But look at the way it is structured. There we have, again, the phosphate sugar thymine, the phosphate sugar cytosine, the phosphate sugar adenine, the phosphate sugar guanine. They've given you a key for that as well, P-phosphate, and sugar. What sugar we're talking about is deoxy. Guys, remember in your tests, in your exams, whenever you're labeling the sugar in DNA, you must use the words deoxyribose sugar, not just sugar, because there are different sugars in DNA and RNA. That stands to reason. Now, again, going back to the location of this guy that we are talking about. Here's a plant cell, an example of a plant cell. And when we look at it, we find that here's the nucleus in this area here. And inside the nucleus, you have the nucleolus, and you have the chromatin network. The DNA is found there. The DNA of the nucleus is found inside the chromosomes. In other words, what happens here is the chromosome is made up of two strands called chromatids. And each chromatid, in turn, is made up of large strands of DNA. And each DNA is made up of smaller units called genes. And it is these genes that we started this topic with. These are the genes that determine what you look like. Not look at, Katlego.
Not
look,
what you look
look
at.
like. In other words, whether you're going to have blue eyes or brown eyes, whether you're going to have straight hair or curly hair.
So it's not whether you have black genes or blue genes, right?
No.
Okay,
That's not.
okay.
It's just different genes code for different things. But we're going to talk about that lots and lots and lots about the blue eyes and the brown eyes under the section called genetics later in the year. So this is where we are at in terms of the location. Guys, why do we put a diagram on? Because a picture says much more than 1,000 words. If I show you the picture and I say DNA's location is here in the nucleus, in the chromosome, already you're seeing it. You're seeing that it must register. And obviously, the mitochondrial DNA will be found there, mDNA. And the chloroplast DNA, cDNA, would be found there. Like I told you, we are interested in this guy, and we are interested in the DNA that's found in the nucleus. That's what we are interested in our studies. A little more on the bases. Adenine and guanine are double-ringed structures, and they are called purine bases. Adenine and guanine are double-ringed structures, and they are called purine bases. Thymine and cytosine, I should actually do that and change the color. Thymine and cytosine are pyrimidine bases. And they have single-ring molecules. They are single-ring molecules. The chemistry, you do not have to go into the details. Just you've got to know that cytosine and thymine are pyrimidines, and adenine and guanine are purines. That's what you need to know. Guys, that's the first part, and we're talking about the structure. We're going to go into more detail with the structure after this break.
Thank you, Aslam. Guys, let's just go for a quick break. I know you need it. I hope your notes are starting to look quite good, because I know that you guys have already commented that your notes are fantastic because of your pens, your highlighters, all the information that we've given you. So you can just do that during the break, but make sure that you don't move, guys. See you just after this. Welcome back, grade 12s. If you've just joined us, we are doing life science for grade 12s. And of course, we are doing the genes today. It's all about the genes today. So, guys, make sure that you are totally tuned in and your full attention is on the page and on your screens. And also, one thing I need to talk to you about. Guys, you need to appeal to your teachers, go to your teachers, do anything you can to your teachers so that they can send us some content, guys. We need some content from your awesome teachers. The more, the better, guys. Anything that you can get, tell your teachers to send it at content@mindset.co.za. That's content@mindset.co.za. Some of you have some great teachers, so make sure that they send us all that they have. Another thing, guys, there is this awesome presenter search going on. If you guys tune in to your grade 12 lesson tomorrow, same time, same place, you get to view the top 10 presenters, guys. Top 15, actually. Top 15, not even top 10. This is so much better. So from then on, we'll choose a top 10. And you guys can see this all happen. Aren't we amazing? Yes, I think we are. Over to you, Aslam.
Thanks, Katlego. Great stuff, guys. I'm hoping you're keeping your body hydrated in this hot weather, and you are keeping those genes flowing. All right. We move on a little further, just an interesting observation. And I want you to concentrate on the information that's on the board, on the screen. So much so used to the classroom, you see,
Yeah.
on the board, even though I use PowerPoint all the time. Anyway, so here they're telling you, and again, a skill involved here. It's not only about the content in this case here. I want you to focus on the skill of drawing a table. Simple. So when we're drawing a table of any data that we collect, we have a heading for the table showing the percentage, notice, percentage of each of the 4 bases, one variable, in the DNA from different organisms. That's your other variable. So on the one end, you got the amount or the percentage of DNA of what? From different organisms. And as you can see, the source, there's the different organisms here. Man, chicken, salmon, locust, wheat. So that's your one variable. And here's your other variable, the percentage of different bases found in the DNA. And just 5 minutes ago, we learned that the 4 bases found in DNA, whether you're a plant, vegetable, Katlego? You think you are? No, you're not at
No, I
all.
don't think
You're
so.
talking back, so that can't be. Whether it's a plant, not you're a plant, sorry, whether it's a plant, whether it's an animal, whether it's a butterfly, whatever, fish, whatever type of animal, all organisms have these 4 bases in their DNA: adenine, guanine, thymine, cytosine. In man, they found this 30.9 adenine, 19.9 guanine, thymine 29.4, cytosine 19.8. All right? Chicken, 28.8, 20.5, 29.2, and 21.5. Now, guys, obviously, I'm not going to read the whole table for you. I'm going to take one more example. Let's take wheat. Take an example of a plant. 27.3, 22.7, 27.1, 22.8. Now, if I had to ask you, what trend do you see in the table, taking the 3 organisms that we have looked at only the 3 organisms that we have looked at, in terms of the bases? What would you notice? You thought about it? You've got an answer? Okay, now we're going to discuss the answer, and let's see whether you are on track. Notice that in man, adenine, 30.9. Thymine, 29.4. In chicken, 28.8, 29.2. In wheat, 27.3, 27.1. And if we take the guanine, 19.9, 19.8 cytosine in man, 20.5 in chicken, 21.5 in cytosine, of cytosine. In wheat, 22.7 guanine, and cytosine 22.8. Now, by now, you should have noticed, and look at the color scheme that I used. I used green for A and T, and I used purple, pink. Colorblind I am. I'm a man after all, so I don't really have much taste for color.
Except you're wearing a nice bright green T-shirt. I hope you can see
Oh,
that.
yeah, that I can see. As long as I'm not red, green,
Yeah.
colorblind, then I can see the
Yeah,
traffic
you
lights.
can see.
Pink and that don't really appeal to me. But I wear pink, Katleho. Okay, so I've used those colors to match. Now, what we're looking at? Notice 30.9 adenine, thymine 29.4. You'll notice 28.8, 29.2. So you can see that there's a close relationship between adenine and thymine. The amount of thymine is very similar to the amount of adenine in each of those organisms. And you can go right down the line, you see the same thing. And the same way it goes with guanine and cytosine. Why is this so? We're going to unfold it as we go along in the fact that DNA, first of all, is a double-stranded molecule. It's made up of 2 strands. And the way the bases are pairing is exactly what we are seeing here. That is why we're seeing this. Adenine base pairs with thymine. And guanine pairs with cytosine. So obviously, the amount of adenine in a molecule should be more or less equal to the amount of thymine, and the amount of cytosine should be more or less related to guanine and cytosine. And if you remember from last year, Katleho, we used 2 letters, or two sets of letters, at and CG. At, adenine always combines with thymine, and cute girl. Cute girl, cytosine always combines with guanine. You can remember any one of these 2. You don't have to remember both. You only have to remember one set. Cute girl, I'm sure most of the guys will remember.
Yeah, most of the guys will remember that, definitely.
So you remember cute girl. And if you remember cute girl, you know that cytosine always combines with guanine. It is only 4 bases. If cytosine only combines with guanine, Katleho, it's obvious, goes without saying, that adenine will combine with thymine. And there we have that story done there. Good. Now we're going into the history, guys. Remember we said location, then we looked at structure, and now we are moving towards the discovery, the history. How do we know about this DNA? I mean, we didn't get up one morning and say, look, DNA, this is what DNA is. It took years and years and years of research to get to this stage. And here we have these little snippets in terms of who discovered what. Firstly was Frederick Griffith, who discovers a factor in disease bacteria that can transform harmless bacteria into deadly bacteria in 1928. So he found a factor. Notice the choice of word here. They didn't give it a name, this thing. They just called it a factor. It was something that caused a transformation in the bacteria. Right? That was in 1928. Later, in 1952, Rosalind Franklin and Maurice Wilkins, they were using X-ray photos, and they made an X-ray photo of DNA in 1952. While these guys were busy, our 2 friends, Watson and Crick, they were also busy trying to unfold the structure of DNA. However, they just couldn't get their finger on the pulse. This guy, Maurice Wilkins, for some reason, he had a fallout with Rosalind Franklin, and he then leaks the X-ray to Watson and Crick. And that was their missing link. They already had other research. They put the X-ray together with the other research, and they then described for the first time properly the DNA molecule in 1953. Notice 1952, 1953. Now this brings about an important concept in science, that of ethics in science. we have to ask ourselves whether this was an ethical thing to do, leaking information to somebody and the others accepting that information and then presenting their findings without acknowledging Franklin's work. Now, how does this affect you? You're doing homework, you're doing assignments, you're doing research. Are you simply cutting and pasting without acknowledging the source? Are you looking for somebody's assignment on the internet and simply pasting it and giving it as your own? These are some things that you need to think about, because that is dishonesty, firstly. Moral. It's a moral dilemma on its own. Apart from that, should you go to university, and even in school, if it is proven that your work or the work that you handed in is not your own, you can lose the entire mark of that assignment, research, or whatever you're handling. And it's not only in life science, it's across the board. And remember, with the technology that is available now, educators, lecturers, and the like can use software to determine whether your work has been Googled and pasted in, or it's your own work. So be careful about that. But leave alone what's the consequence. Think about the moral dilemma with it. So think about that. So these guys then do that, and because of their work, they were given the Nobel Prize in 1962. By this time, Rosalind Franklin already had passed away. And the Nobel Prize was not handed out to anybody posthumously. That means posthumously after you die. If you die already, they would not give you that award. So she played an important role with the discovery of the DNA, but she was not recognized at that time. So those are some of the things you must think about. Okay, now we are talking about what the table alluded to, and this is the complementary base pairs are joined. There's your one strand, strand A, strand B. Notice you have your phosphate, sugar, etc. Here's cytosine, guanine, thymine, adenine, guanine, cytosine. So that is how the molecule is combined. Over and above that, you'll notice there's some dotted lines in between the nitrogenous bases. These dotted lines represent rather hydrogen bonds. And not just hydrogen bonds, but weak hydrogen bonds. Now, what's the significance of this being weak? I'm going to ask you this question a little later. I want you to think about it. I'm telling you that the bond between the two strands is weak. And I'm going to be asking you a little bit later, why? Why are these bonds weak? Why don't we use super glue? In other words, hypothetically speaking, super glue meaning having a stronger bond there. You think about that. All right? And here we have it. Two polynucleate strands lying, nucleotide strands lying side by side. In other words, this is what the molecule looks like. But in reality, they don't stay in this form, but they form what we call a double helix. Double, easy, two strands. And helix, as you can see the shape, spiral. So if we say DNA is a double helix, we mean it is made up of two strands which are wound around each other, like a coil, like a spiral staircase, whatever you want to remember it by. But the point is that it is helical, helical. What are the functions of DNA? So this is the next step. Let's go back. Our first step, what is this DNA we're talking about? Next step, where do we find the DNA? Third step, what is it made up of? Fourth step, what are its functions? And what are its functions? Very simple. You learned this in grade 10 already. Carries hereditary information. Hereditary, things that you can inherit. In other words, things that can be passed on from parents to children. Carries hereditary information. And it contains coded information for protein synthesis. This is absolutely crucial. The second function as well. It's absolutely crucial because DNA is the starting point for protein synthesis, as we will learn in later lessons. And with that, I think you've got a mouthful. Back to you, Katlego.
Thank you, Aslam. So, guys, we are going to take a break. This has been really awesome, guys, because you guys are so involved. Your questions are great. Everything is great. How you guys are interacting with each other and helping each other, that is fantastic, guys. You only learn more from helping someone else. So make sure that you keep helping people and you keep asking for help. Ask us, guys. That's what we're here for, after all. See you after the break. Welcome back, guys. You guys are really, like I always say this, but I really mean it. You guys have really been great today, guys. I'm loving how you guys are actually also answering questions. It's great to see that you guys are actually awake. I know it's just the beginning of the year, but please keep it up right till you write your end of year exams or finals, if you want to call it, because I want to see your marks as well. So welcome to grade 12s. We really want to know about your updates. Okay, guys, another awesome fact. Remember that top 15 of our presenter search that I was telling you guys about? It gets greater. You guys will be voting for your top 10, guys. So keep your eyes posted on the page tomorrow so that you guys can vote who you think is the best presenter. Isn't that awesome, guys? I know it really is, and I know you guys love getting involved. So make sure that you are on the page 100% all day long, guys. We love to see you here. Don't get bored. And when you do, just join us on the page, okay? Over to you, Aslam.
Katlego, thank you very much. Guys, keep your eyes on that page. We had one question, Katlego.
Yes, well, quite a few people have asked this question now. They ask, do twins have the same DNA?
Wow, that's an interesting question. And guys, I'm glad that you're asking these questions already, because this question is linked to so much that we're going to do. The question the learner is asking is very simple. Do twins have identical DNA? In other words, do they have the same DNA? Simply put. Now, first of all, we have to understand that we get two major types of twins. The first set of twins are called identical twins. Okay, identical twins already gives you an answer. Identical twins come from the same sperm and the same egg. Keep that in your mind. And the other set of twins that we talk about are fraternal twins. Fraternal. These twins arise from two different sperms and two different eggs. That means there will be two sperms and two eggs got fertilized at the same time. These, obviously, because they are two different sperms and two different eggs, then obviously they would have different DNA. So the identical twins, Katlego, to answer the question, have the same DNA.
Okay, that makes sense,
because
right?
they came from one fertilized cell. There was one sperm that fertilized an egg cell. While development was taking place, the egg splits in two.
Okay.
So from one cell, we got two separate human beings, or, well, any organism that were twins, identical, developing. And obviously, the DNA was in the sperm, and the DNA was in the egg. That same DNA would be found in each of these twins.
Okay.
In the second case, two separate eggs and two separate sperms, they would have different DNA. Their DNA will be...
look different.
They would look different. It could be a boy and a girl. And obviously, Katlego, many people ask me, they look at a brother and a sister as twins. Right? And they look very similar.
The same, yeah.
And they say, are they identical? I say, come on, guys.
Come on,
look
one's
a little
a
bit
boy,
lower
one's
down.
a girl. Okay,
They're looking too high up. They're only looking up to here, you see. And they're not looking down. And if they look down, then they know they can never be identical. They are actually complementary.
They are complementary, not identical.
Not identical. Okay. So that answers that question. I'm hoping you all click it. And please keep that in your mind, because when we do DNA fingerprinting later in the year, you will have to use that information that we have just discussed. We move on now to DNA replication. To replicate, to make a copy. Make a copy. And because we're using the word replicate, make an exact copy. Identical, if you want to use the word. This is crucial. Right, so we're explaining what it is, where DNA in the original cell has to produce an exact copy of itself. By the way, lots of people, friends and family, and also people that watched last year, they asked me, Katlego, that
Yes.
for today, I must wave.
Okay, so
So
that they
I'm
know.
waving to
okay.
all of you
I'll
that
also
wanted
wave.
me to wave. So Katlego is also wave. One guy actually told me, pull your ear, so I'm doing that also.
Oh my
too.
goodness.
Okay. All right, so have to please the fans.
Yeah, I know what you mean.
Okay. All right. Where do we find this? Where does this take place, rather? In the nucleus, because the DNA that we are talking about is in the nucleus. So where will it replicate? In the nucleus. When does it do so? It would do so during interphase of cell division. Now, this cell division, you may say, is going to come in the next chapter. However, guys, don't lie to yourself. You've done cell division in grade 10, mitosis. Yes, we do meiosis after this chapter in grade 12, but you've done mitosis, and you know the 5 phases: interphase, metaphase, interphase, prophase, metaphase, anaphase, and telophase. The interphase was the phase between two phases. And what happened during that phase? DNA replication, and that's what we're talking about now. Why does this take place? And why could also mean, what is the significance? Another way to ask why is, what is the significance? Why does it happen? This is what significance means. If I ask you, what is the significance of something? Why is it there? Or why does it happen? Remember that. Again, people, keep tuned in to the little words and the little phrases that we are talking about in terms of skill. Keep that in mind. Grade 12 is more about fine-tuning, less about content. The content is important, but you need to know what to do with the content. So listen very carefully. Why? So that the genetic code is passed on to each new daughter cell formed during cell division. The cell is dividing, it's making more cells. And if the DNA does not have copies, how can one cell then form two cells without making a copy of the DNA? Which cell is going to get the DNA and which cell won't get the DNA? So when DNA replication takes place, the two cells will each then have identical copies of the original DNA. That's what we are saying. That's why it takes place. Now, there were two chains of thought in this whole process, and this is just a rough representation of that. If this is the original molecule of DNA, and conservative replication says that the molecule that comes out of this comes from this whole molecule on its own, and you get this here. The second chain of thought says semi-conservative replication, that when DNA replication takes place, the two strands separate, and each strand makes another strand. But yes, because they are complementary to each other, the two resultant strands will look identical to the original. And here we say that this is semi-conservative replication, and the second theory is the one that is more accepted. Let's look at what actually happens. If this is a DNA molecule, the first thing that happens is the DNA molecule unwinds. The DNA molecule unwinds, as you can see. It's unwinding. It's separate. Unwinds means the strands separate into a ladder-like form instead of a helix or a spiral. The enzyme, and Katlego, there were some questions asking how do the strands separate.
Yes.
This is the answer. If you ask the question, how do the strands separate, this is the answer to that. The enzyme DNA polymerase unzips the DNA by causing the hydrogen bonds to break. So an enzyme is important in this process. You do not need to know the name of the enzyme, but you need to know that an enzyme is involved in breaking up the molecule, or rather splitting the hydrogen bonds. So what happens? And this year we say the word is used is unzip. First word we use, unwind. Second word we're using, unzip. And, oh yes, wow. I always don't have a zip when I want to show this.
Oh, now you have a zip.
Now I have a zip in front of me where I don't have to actually reveal anything. Okay, here's the DNA molecule.
Yes, that's the point.
And
it
it unwinds and it unzips. I actually brought the genes just for that first part.
Oh, okay,
But
now
they-
they're
it
useful.
helped me with something else.
Yeah.
Unzip simply means, guys, that the hydrogen bonds break and the strands separate, unzip. As you dropping that zip, the two sides of the zip will separate from each other. Why you do that, that's another story. Okay? So that's unzip. While that is happening, in the nucleus, there are many nucleotides that are freely floating in the nucleoplasm. And these nucleotides join up with the two separate strands in a particular fashion. What fashion? A will combine with T, and C will combine with G. Cute girl. At. That's what's going to happen. So on each side, this is going to happen. So in other words, what has happened? The two strands separate. Yes, strand 1, strand 2. Each one acts as a mold, M-O-U-L-D, or a template on which a new strand forms. As you can see, there's the templates there and there. And on each one, C-G, T-A, C-G, A-T, G, C-G, T-A, G-C. That's what's happening. So the nucleotides will combine. Again, enzymes will be involved in helping to make this bond. Just as enzymes were involved in breaking the bonds, enzymes are also involved in making the new bonds. And what happens is we get, and then what will happen thereafter, the template will form, and I will use the word 5, I would use the word 6, re-zip there, re-zip actually with 5, and 6, rewind. I want you to concentrate on those words. Unwind, unzip, re-zip, rewind. And the last word will be enzymes because you're going to involve in 5. So what do we mean by re-zip? They attach again. What do we mean by rewind? They form a helix again. So in pictures, that's what we see. Now, let's look at the words. So the first thing that enzyme unwinds, we got that, or rather the DNA unwinds. The enzyme DNA polymerase unzips the DNA. In other words, the hydrogen bonds break. Each single strand acts as a template, that's another word. Free nucleotides in the nuclear pair up with their complement, this is a re-zips. And each single strand becomes a new double strand, and they rewind. So can you see that just by learning these words, unwind, unzip, re-zip, rewind. Just these 4 words tell you the whole process.
Yeah, that's fantastic.
Guys, I want you to remember these words. And in between, you can put the word enzyme, and you can put the word template. I think
It's time for that break, eh? Okay, guys, I know right about now, you're probably hating us having so many ad breaks. But just for those people who quickly need to get a glass of water or need to use the loo or refill their pens or anything like that, this break is for you guys. Join us right after this. Welcome back, Mindsetters. Most of you guys are chatting on the page about how life science has just become your favorite matric subject. Well, I'm happy for that, and I'm sure Aslam is so happy to hear that, because obviously life science is his favorite subject, so now he can share it with you guys. So now you guys can be more motivating and even post some nice pictures, guys. There's some nice motivational things that you guys are talking about. Do that, guys. This page is for you. Don't forget, this page is just for you. You don't even need to share it with anyone except obviously your peers. But I mean, it is for you. And don't forget that Mindset website, guys, with all the notes and the schedules. Check it out, guys. Click on it a few times, explore it, and see what you can find. There's some awesome stuff on that website, guys. Don't miss out. I'm warning you, don't miss out. Over to you, Aslam.
Thanks, Katlego. Guys, welcome back. I'm hoping you're refreshed and you're at the end. And it's not just a favorite subject. It's the most important subject. Life science. Life science. The science about life.
There's nothing more important than that, eh?
Remember, I asked you a question, guys. I asked you a question about why do we have this hydrogen bond that is weak. Katlego says many people have answered it. You want to share some of those answers with me?
Okay, sure. I'll share some. I've combined
I think most
them.
of the
So
same.
Hope, Mohosi, and Tulani, you guys answered first. They say that the bond is weak so that they can break easily or break down when forming RNA.
Okay, they're jumping 3 steps ahead of us.
You know our mindset.
That's Hope, Mohosi, and Tulani.
Hope, Mokhosi and Tulani.
Hope, Mokhosi and Tulani, you're on the button. But in terms of what we did today, you gave the first part of the answer. Absolutely correct. It is weak so that it can easily break or break easily. And in our case, because we're not talking about RNA yet, during replication, when unzip. In other words, when the molecules unzipping, what is happening? The weak hydrogen bond is breaking. If this bond was tough, it wouldn't be able to break, and we wouldn't be able to make copies of our DNA. And Hope, Mokhosi, and Tulani hold on to that thought for RNA as well, because we're going to come back to RNA in later lessons, and we'll talk about the RNA as well. Thanks for that. Okay, guys, now we've learned so much. Let's see if we remember some of it. In your section A, the first question is always multiple choice. 1.2 generally would be provide the correct biological term. And the examiners and markers are sticky. They want the proper biological term, not just a common kitchen term. They want the proper biological term. So we're starting today with some of those. Correct biological term for the following: the base that pairs with thymine. Yep, you're right, that is adenine. And how we know that? At our story. The building blocks of proteins. Okay, we haven't come to this stage yet, but let's just write it down for you. Amino acids. We'll talk about this when we talk about RNA. The bonds that hold the two polynucleate strands of a DNA molecule together. We've just been answering a question on that, and that is the hydrogen bonds. And I would be a little more finicky and say you must write the weak hydrogen bond. You don't even have to write bond because the question says bond. Weak hydrogen is good enough. Repeating units, monomers, that form a nucleic acid. And that is, I spoke about the bricks on the wall. Yes, that is nucleotides. Phosphate, sugar, nitrogenous base. The sugar that forms part of DNA, that's your clue, DNA would be deoxyribose. You don't have to write the word sugar because sugar is already in the question. The shape of a DNA molecule, you will say spiral, or you can say helix, or you can say double helix. They only asked for the shape. So we're talking of the spiral nature, the helix nature of the DNA. So can you see that's your one-word answers. We move on now to the structure of DNA. The diagram below represents a part of a molecule. Study the diagram and answer the questions that follow. There's a diagram there. They're giving you A is adenine, G is guanine. By now you should know that. No, even if they don't give you the key, A is adenine in a DNA, G is guanine, C for cytosine, and T for thymine. And they've given you the molecule there, and they're saying identify the molecule above. Because we did DNA today, it's easy, but in exams you've done DNA, you've done RNA, so it's difficult. But the fact what you're going to look at, you're going to look at it has two strands, double strand, so therefore this is DNA. There's nothing else in the diagram that tells you anything else, so you can't use anything else there. Label the parts numbered 1 and 5 respectively. 1 and 5. Now, here, there's nothing here, there's nothing here. How do I know this? Wow, what do I do? I'll come back to that. Let's do 1 first. 1 is easy, it's the bonds. So this is the weak hydrogen bonds. 5, now look at 5, look at the shape of 5, and look at the shape of this molecule. Look on this diagram, find another molecule that looks like this, just like a jigsaw puzzle, and there you have that thing there. And therefore, if that was G, then this must be cytosine. I'm just writing cytosine, cyto, you'll know the rest. What is the collective name for part number 2, 3, and 4? Obviously, without even looking at 2, 3, and 4, I can tell you when I use the word collective in a DNA molecule, it would be a nucleotide. Okay. What is the significance of this molecule being able to replicate itself so that the daughter cells have the same chromosome number and the same DNA as the parent? Let's look at this one quickly. Let's say when a sample of DNA was analyzed, 31% of the bases was to be cytosine. What percentage of guanine would you expect? For every cytosine, there must be guanine. So if 31% Katlego was cytosine, then
Then
putting you on the spot.
70, what?
no,
79?
cytosine and guanine must be the
69.
same.
Oh, cytosine and guanine, so
So if 31, it must be
31 as
31.
well.
Because they are the same. You see, Katlego, I quoted there, that one.
I know, you caught me there.
Okay, so there's one more question on calculation, but it's the same type of a question as this. If you ask for thymine and adenine, a type like this, you got to minus. Katlego.
Thank you, Aslam, for an awesome show. And thank you, Mindsetters, for joining us today. You guys are absolutely amazing. I've been saying this all show, so you guys know who you are. And I want to quickly make a shout out to 2 Mindsetters who actually got distinctions for life science. They say, thank you to Mindset. This is Tumi Faith Moy and Daphne Khanse Sentimile. Guys, well done. Hey, you guys make us proud. And you motivate every other life science mindsetter, guys. So well done, guys. From us at Mindset, thank you for joining us, guys. And to Macmillan, thank you for sponsoring our show. Bye-bye, guys.